Okay, makes sense. We're back at it. Okay, FRQ. Number 1. Red king crab, Paralathidus conscatus, inhabits shallow coastal waters in the northern Pacific Ocean that can vary in temperature from 1 to 13 degrees Celsius. The body temperature of the crab is typically similar to that of the surrounding water. In order to study the effect of temperature on crab metabolism, scientists purified the enzyme PFK from red king crabs. PFK catalyzes the reaction early in the glycolysis pathway, which is shown in figure 1, which just shows that. So, the scientists determined the rate of the PFK catalyzed reactions using a constant amount of PFK and increasing concentrations. Increasing concentrations of the substance F6P at 15 degrees Celsius and 25 degrees Celsius. To determine whether the reaction rate is affected by the nucleotide AMP, the analyses were additionally performed in the presence of 0.5 millimeters AMP. In a second analysis of PFK activity at different temperatures, the scientists determined that the concentrations of citric acid in the reaction mixture affects the rate of the reaction at 5 degrees Celsius, 15 degrees Celsius, and 25 degrees Celsius, figure 2. In the presence of a constant amount of PFK, citric acid is the first metabolic intermediate produced by the Krebs cycle. All reactions were performed in the presence of 1 millimeter F6P and 0.5 millimeter ATP. Figure 3, effect of citric acid on the rate of the... Just a description of the graph, which I assume you have access to. So, question A, determine the role of ATP in the reaction, shown in figure 1. I wrote, the PFK catalyzed reaction is glycolysis, which has investment phase. In this phase, ATP is given. And then I said PFK catalyzes during the energy investment phase. So I was supposed to include how ATP provides both the phosphate group and the energy required in the reaction. Well, I did imply that energy was, I didn't write this, but first off, point, I got that identified glycolysis. Identified glycolysis. While I implied the energy was input, I did not state it. Our correct answer would have read, ATP is necessary because it requires both the energy and phosphate group required to convert F6P to FBP. Moving on. Let's move on to question B. So, question B asks, identify an independent hypotheses for the experiments. Shown in figure 1, scientists claim that in the absence of AMP, PFK of red crab substrate more tightly at the low temperatures than it does at the higher ones. Support the scientists' claim based on the data provided. Predict the effect on the rate of the reaction catalyzed with PFK. In the experiment shown in figure 2, if increased concentrations of FBP are added to the reaction. My response read. Apologies. My response read, independent variable is temperature. Adding AMP leads to an initial increase in the rate of reaction before a plateau. It will be 4 times greater at 25 degrees Celsius. Increasing F6P makes sense because it is the starting materials of PFK catalyzed into PFP. Thus, increasing allows for continuing reactions to take place. This AMP causes increases in comparison to no AMP. The matter is lower than if there were no AMP. We'll measure how much it takes. My response had an acceptable independent variable temperature. I also concluded the science degree culture to determine when it would be level off. However, I forgot to pinpoint that below 0.15 millimeters, the reaction is more rapid and that higher is slower. Also, I incorrectly calculated the fold increase. A correct answer would have read an independent variable is temperature. The scientist would have increased the concentration until the reaction levels off. Below 0.515 is less rapid and above is more rapid. And I would calculate it to be 11. My answer was fully right. Don't need to go over it at all. Okay. Question D. Question D reads, while crustaceans such as crabs are kept in seawater with lower than normal pH, ATP requiring pumps that maintain ion gradients in the animal's body significantly increase their activity in comparison with animals with normal levels. Explain how the increased use of ATP by these pumps will most likely affect the ability of the animals to carry out other metabolic reactions, such as that catalyzed by PFK. Hemolymph is the fluid in which the red king crab's body surrounds its cells and is typically slightly hypertonic to the seawater in which the crabs live. When adult crabs were removed from their native environment and placed into seawater solutions with gradually decreasing salt concentration, their body weight increased. Explain how the crabs gained weight during this procedure. Okay, my answer, I said, using more ATP, there means less available elsewhere, more hemolymph, other parts have less ATP to use. I included that using more ATP means there's less use elsewhere, however, did not include the water moved into the crab. The full correct answer would say that increasing ATP usage leads to a drought elsewhere and that water enters the crab because it has higher solute density there. Moving on to question 2. Don't go back to Harlem. It doesn't matter anymore. Okay. Many species of bacteria grow in the mouths of animals and can form biofilms on teeth plaque. Within plaque, the outer layers contain high levels of oxygen, and the layer closest to the tooth contain low levels of oxygen. The surface of the tooth is covered in a hard layer of enamel, which can be dissolved under acidic conditions. When the animal breaks down the bacteria in the plaque, it can extract nutrients from the tooth and cause cavities. Certain types of bacteria, Streptococcus mutans, thrive in the innermost anaerobic layers and associate cavities. Other types, Streptococcus, compete with S mutans but are unable to thrive in acidic environments. Question A. Identify the biochemical pathway S mutans uses for metabolizing sugar and describe how the pathway contributes to low pH in the inner layers of plaque. My answer was, don't go back to Harlem. It doesn't matter anyway. I said that pathway of glycolysis, low pH because H plus is let off as a waste product by joining oxygen. I correctly identified glycolysis, however, did not identify that there would be a pyruvic acid change. A quick answer glycolysis is an effective pathway with pyruvic acid production change. Part B of question 2, I got fully right. Moving on. Question. Moving on. Question 3. Oh my god. Non-cyclic electron flow are two major pathways, and cyclic electron flow are two major pathways of the light-independent reactions of photosynthesis. In non-cyclic electron flow, electrons pass through photosystem 2, then components of chloroplast electron transport chain, and photosystem 1 before finally using Mg plus and ADPH. In cyclic electron flow, electrons cycle through photosystem 1 and components of electron transport chain. Question A, I got fully right. Question B, I wrote, cyclic use of electron carriers, increasing the number of electron carriers will increase its rate of being used. I needed to include there's less NADP. Right answer would say that there's less NADP, which means less carriers to accept electrons, so they go to cyclic pathway instead. Moving on to, don't go back to Harlem. It doesn't matter any. Sorry, I don't know if that just happened to you. My thing just bugged out. Don't go back to Harlem. Question C. Using rice plants, scientists examine the effect of the mutation. I'm going to combine question C and question D because they're pretty connected questions, so just so you know. Using rice plants, scientists examine the effect of the mutation that results in the loss of protein CRR6. CRR6 is beneficial to wake up back predict the effect of the mutation, the rate of biomass, dry weight accumulation, and justify your prediction. I said dry weight. I said increase. As the dry weight increases inversely with cyclic electron flow, while only for system 1, thus less citric weight means more dry weight. Question 2 was wrong because it said increase instead of decrease, right after say it would decrease. And it was wrong because not enough ATP because it was relying solely on the cyclic. And now I'm done. Ning Ning is the Maknae.